And these are the most abundant, the most abundant branch of the vasculature, also probably the most meaningful, right? This is where we have the exchange. Remember the circulatory system, blood flow, the whole sole purpose of that is to oxygenate our tissues and to remove metabolic waste products, including carbon dioxide. And so at the level of the capillaries and the capillary beds, this is where that magic actually happens, that transfer of nutrients and oxygen and the removal of waste products from the tissues. So this is really where the combination of what the purpose of the circulatory system, that combination is happening here at the capillaries. We'll talk about the capillary anatomy first, so the different types of capillaries that we have in the body. Then we'll talk about ways that we can control that locally. So again, a few intrinsic mechanisms of controlling flow to capillary beds. And then we'll talk about the actual process of exchange. And this is where we'll get into the pressure gradients and calculating the different gradients there and determining whether or not we have transfer into the tissues or transfer back into the capillaries. Remember, there's exchange happening there. And so there's that bidirectional movement of things from either side. Okay. Now, so this is the site of the capillary exchange. This is where we have the smallest diameter. And so this is really important. This is going to provide a small barrier and allow for diffusion that we just described, right? That movement of oxygen, nutrients, and those metabolic byproducts. And so the diameter that we see here is really, really small. It's 5 to 10 micrometers. And make sure that you have this correct in your notes. So in a previous version, this may have been millimeters. And I think I went in and corrected that, but just make sure you have it correct in your notes. So it's not millimeters. Millimeters is way too big. For the capillary, it's actually 5 to 10 micrometers in terms of the diameter of the capillaries. The walls here look a little bit different from our arteries and veins. It's one cell layer thick, which is ideal for diffusion. So there's a very small or minimal diffusion barrier. And we also see the greatest cross-sectional or total cross-sectional area. In other words, these are the most abundant branch of the vasculature. And so if we took the total cross-sectional area of all of the arteries, all of the arterioles, all of the capillaries, the cross-sectional area that we see in capillaries would be the largest because there are just so many capillaries that are in the body, okay? And while we're on this point, I kind of want to touch on why, although we have smaller lumen in capillaries, we don't see as much resistance in these capillaries as we saw in the arterioles. This is because of the vast network of capillaries that we have. In terms of the total cross-sectional area, it's much wider than what we saw with arterioles, although the individual lumen of these vessels are smaller. There are just so many capillaries that it winds up being much less resistance than we see in the arterioles. This is also where we see the slowing of the flow, right? The slowest flow. This is ideal for exchange as well. So we don't see that rapid movement of blood kind of flowing through the way we had in arteries and arterioles with that high pressure, because here we really need to slow down in order for that exchange to happen, right? Remember, exchange is happening in both directions. There's arterial, it's not arterial, but there's exchange of not oxygen and nutrients moving into the tissues and other things moving back into the vessel as well. So in order for that exchange to happen, there needs to be enough time. And so there needs to be a slowing of the flow as it moves through this branch of the vasculature. Now there are 10 to 40 billion capillaries throughout the body, so lots and lots of capillaries. The total surface area is about 600 meters square. So think about the size of a tennis court or so. Most of the cells within our body are really close to a capillary or capillary network, within 1 millimeter of a capillary or capillary network. This is why even if you prick your finger, prick your skin, the tiniest of cuts or injuries to the integument or to the skin can produce a little bit of bleeding, because the capillaries are found abundant throughout the body and are usually 1 millimeter within most of our cells. There's also 1 millimeter in terms of the length. So there needs to be, again, enough length to facilitate the time that it takes for that exchange to happen effectively. Now, in terms of the lining of our arterioles, we see So we talked about it being one cell layer thick. So we don't have the bulky, smooth muscle. We don't have the connective tissue that we saw in other areas. All we see are endothelial cells, right? Just that one cell layer of endothelial cells lining the outside of this vessel. And in between those endothelial cells, there are some pores. And the pores allow for the protein-free plasma to move throughout, out of that capillary and into the tissues. And when we say protein-free plasma, these pores usually, in terms of the most common type of capillary, there are different types, but the most common type, which I'll talk about in a minute, are continuous capillaries. And so the opening between these endothelial cells are really small. And while they allow the movement of plasma for the most part, they do not allow the movement of large cells or the movement of proteins. And so what we really want to underscore here is that protein-free plasma is what is moved in between those pores that line, or those pores that are in between the endothelial cells that line the capillaries. We also want to talk about this idea of the capillary bed. So this is a network of capillaries, and we don't really look at or talk about capillaries on an individual level because they're just so small, so minute, so many of them, but we really refer to the network of capillaries, which we call a capillary bed. And so we look at arterioles that flow from arteries into capillary beds. And one arteriole is basically the regulatory point that can manipulate how much blood goes to this capillary bed or whether or not blood goes away from the capillary bed. Okay, so we can regulate in the arterioles how much blood is flowing to the different capillary beds and not necessarily an individual capillary as such. So we're looking here at the total cross-sectional area, just kind of catching my bearings here. We're looking at the total cross-sectional area of capillaries. And what we're seeing is that as we get to the capillaries, because there are just so many capillaries, the total cross-sectional area is the greatest. Therefore, the velocity of flow is the lowest. And a practical way to explain this is think about a river that's running under high pressure that dumps into a lake, okay? So as we get into that more vast area, that more vast network of capillaries, the flow slows down, so we lose the velocity here. And then we also see that this has a greater cross-sectional area in total, okay? And this is, again, ideal in terms of allowing that time for the exchange to take place. So we see the most abundant or the greatest cross-sectional area for capillaries, and then we also see the slowest velocity of flow to allow for that exchange. Okay. Let's speak here about the different types of capillaries that we have in the body. So we have three main types of capillaries. We've got continuous capillaries. These are the most common or the most abundant. And in between the endothelial cells that line these capillaries, we can have movement of small, uncharged, water-soluble molecules moving right through, okay? We don't have the movement of cells and we don't have the movement of bulkier structures like proteins. In fenestrated capillaries, we have larger gaps in between those endothelial cells. So they form these pores or these windows or openings that we call fenestrations. And these do allow proteins and in some instances, blood cells to move through those tiny pores. And so continuous capillaries are the most abundant that we see throughout the body. Fenestrated capillaries are a lot more rare. We do see them in areas like the endocrine system. So this is how hormones can move in and out of the blood and into tissues and so on. We also see them in parts of the kidneys where we talk about the glomerulus that actually filters blood. So they need to have like slightly larger openings or fenestrations to allow for that movement, that filtering of blood. And so they're a lot more rare than we find the continuous capillaries. The third branch of capillaries are the discontinuous capillaries, also called the sinusoidal capillaries, okay? And these are, again, even more rare, and these are found in areas like the bone marrow and sometimes the liver. We see the discontinuous capillaries, and these have even larger openings, right? Things can just freely flow through the openings or the windows in between the endothelial cells for these discontinuous capillaries. Some of the examples here are the bone marrow. So think about the production of erythrocytes or red blood cells in the bone marrow. They need to be able to escape to get into the blood. And so having a discontinuous or more freely open flow between bone marrow and the capillary network is ideal in that instance in terms of that movement of red blood cells.